been obtained by a transition from A to A here, or from A to C here, or from A to G here, or from A to T here, right? So I've used the OR that explains the summation sign here, right? Because it could either, A could have either appeared here by a transition from A to A, C to A, G to A, or T to A, and then at the same time, a similar transition must have happened on the other side. So this is like an AND, that's why we're multiplying here. And here we're considering a sum over probability of going from A to A to C along this branch, of course, A to G and A to T. And with that, we have computed our first value in this conditional likelihood vector here by summing over the transition probabilities from A along branch BI to the conditional likelihood vector entries in the left child, and then multiplying it with the sum of the transition probabilities from A over Bj to the respective entries in the right child here. So that's what we actually do at the smiley here to fill up the values, or to fill in the values in this of this green conditional likelihood vector here. Then we continue our post-order traversal here, and on the way back to the root, we then fill in those conditional likelihood vectors here, because we know that, well, if we're on our way back to the root, it's the last time we visit this inner node, the corresponding left and right child vectors will already have been computed. Right, then in the very end, at the virtual root, we then just sum over the logarithm of the per side likelihoods to obtain the overall log likelihood. Right. One important thing to keep in mind when we're doing those traversals is that, well, as we approach the root, since we're multiplying probabilities all the time, let me go back here, that's where we're multiplying probabilities, right? So this is kind of nasty, because the more probabilities we multiply, the smaller our values will get. So this means that in general, like in this very abstract representation, the closer we get to the root, the values in the conditional likelihood vectors will get smaller and smaller and smaller. So at some point, they will get so small that they cannot be represented at the on the computer anymore. So this actually needs to be handled, and what we do here is that we apply a technique that is called numerical scaling. So if those values get too small, we will multiply them with a large number to scale them, and then somehow try to undo this multiplication or the scaling operation at the root once we compute the overall log likelihood value. Okay, another thing to mention here, of course, in order to get the maximum likelihood value of this specific tree topology, we also need to optimize our branch lengths. So we need to optimize each branch length individually to maximize the likelihood. And so this is how that works. So we select a starting branch here, we start optimizing it. So essentially this means that we place the virtual root into this branch here, and then start altering the branch length, and then pick that branch that optimizes the likelihood. And so another important thing here is that we need to synchronize the computations at every branch we visit. Then we go to the next branch, optimize its branch length, etc., traverse the tree in a certain order, and then, because a change of this last branch length we optimized here may then affect the values of the other branch lengths, we actually also iterate over the tree. So we'll start again at the starting branch, readapt the branch lengths, and do so a couple of times until the branch length optimization converges. And this is typically done by saying, okay, so now between iteration I and I plus 1, the log likelihood of the overall tree has just changed by a very small fraction, by an epsilon. So this means that we stop. Another thing we need to do is to optimize model parameters, of course. But we don't need to really unlike with branch lengths, we don't really need to re-estimate model parameters for every tree topology that we visit, as long as the tree is reasonable, meaning that it's like not a totally random tree. So this is something those, unlike the branch lengths, those model parameters or the optimization of their values is nothing that we need to worry about that much. Okay, so here are the basic operations just as a summary, like if I'm going to implement that on a computer. Here are the basic operations of maximum likelihood. So we need to compute a conditional likelihood array or a conditional likelihood vector at an inner node. We need a function to compute the likelihood at the virtual root. We need a function to optimize a branch length for a given branch. Then we need to optimize a function. We need to have a function to optimize all branch lengths on the tree. And we need one or more functions to optimize the other model parameters like the rates, the base frequencies, the alpha parameter of the gamma model of rate heterogeneity, etc., etc. Right, so here's a key question. Why are Bayesians lazy people? So the key thing in maximum likelihood, when implementing maximum likelihood programs, is that getting those optimization routines to work properly or to work well is very difficult and tricky, right? So this is like the numerical challenge or the implementation challenge here. And with Bayesian programs, we're not doing any optimization, right? So we're just doing some sort of targeted random sampling, meaning that we don't need to build any optimization functions when we're implementing a Bayesian MCMC method. So that's why Bayesians are really lazy. Right, so this is the answer to my question, right? So Bayesian programs only require those two operations, right? Compute conditional likelihood array at an inner node and compute the likelihood at the overall likelihood at the virtual root. And that is basically all they need, and the rest is just handled by the proposal mechanisms. Right, so a couple of notes about rate heterogeneity among sites. We all know that this is a true biological phenomenon, that different sites or columns evolve at different speeds, and we need to accommodate this in our models. So this is the gamma distribution that we typically use, and so it has this alpha shape parameter that gives very different shapes to this curve. So typically like an alpha of 0.5 means high rate heterogeneity. You can see that this distribution is kind of very broad, whereas a large value for alpha means low rate heterogeneity, meaning that this distribution here is very kind of narrow. So what we're doing with this gamma distribution, given that we've estimated our parameter alpha, is that we essentially integrate the likelihood over this entire curve, right? So that's what we basically do. As I said before, just as a reminder, so small alpha means a high level of rate heterogeneity, and a large value of alpha means a low degree of rate heterogeneity. As I said, what we do is that we integrate the likelihood over this gamma curve, and we do so for each site independently. Evidently, this is a continuous integral which we cannot calculate on a computer. So what we do is that we approximate, we do a discrete approximation of the integral. So we select specific discrete rates and then just sum over the likelihood values at those discrete rates to approximate the integral over the gamma function. And so here's like an abstract view of this gamma function. So typically, if we assume 4 discrete rates, if we use 4 discrete rates, which is like the default case in most programs, then basically we need to calculate the likelihood or fill in conditional likelihood vectors in our tree for each single one of those 4 discrete rates, and then the overall log likelihood is just an average over the log likelihoods over the rates, right? So every rate contributes 1/4, because we have 4 rates, so 1/4 contributes equally to the overall likelihood. So why am I showing you this? Well, if I use a gamma model of rate heterogeneity with 4 discrete rates, this means four times higher memory consumption because I need to store conditional likelihood vector entries for each of those 4 discrete rates. Here's another way of looking at it. The gamma model of rate heterogeneity with 4 discrete rates. So like the key mistake or the key misunderstanding about this is that people may think that, well, each site may only be assigned one of those 4 discrete rates, right? But this is not what we're doing. So we don't have any function or any good way to really assign those rates to individual sites. Instead, what we're doing is that we're assigning each of those 4 rates to each site, and then the idea is that, well, the rate that fits this site best will also contribute most to the overall likelihood of this site, right? So this assignment is somehow taking place implicitly by the fact that the rate that contributes most to that best fits the site contributes most to the likelihood. Okay, so now we're actually in a position to calculate the memory requirements of a maximum likelihood, or generally likelihood-based analysis, because the same also applies to Bayesian phylogenetic inference. So if we think about memory consumption, the question here is, what's the accumulated size of all conditional likelihood vectors in our tree? Because as I said before, the memory we need to store the conditional likelihood vectors really dominates our memory requirements. So first, we need to think about the number of conditional likelihood vectors. So if I have a tree with n sequences, an unrooted binary tree with n sequences, I have n minus 2 inner nodes. So I need to store n minus 2 conditional likelihood vectors in the best case. Then I need to know what the number of entries per conditional likelihood vector is. This is just the alignment length, right? So the number of those cells here is the alignment length. Then I need to know how many states I'm storing per likelihood vector entry. So here it's 4 states because this is a DNA example. Then I need to take into account the impact of the rate heterogeneity model I'm using. So here we're not using rate heterogeneity, so the factor here is 1. If we use a gamma model with 4 discrete rates, the factor will be 4. And then finally, I multiply that with 8 bytes, so the number of bytes I need for storing a double precision value, so an arithmetic approximation or representation of the real numbers on a computer system. And that's it. Right, so next step is to talk about parallel likelihood calculations. Okay, so now let's continue with parallel likelihood calculations and how do we do that at an abstract level. So we have different levels of parallelism available when we're doing likelihood calculations. One level of parallelism is what we call embarrassing parallelism, so something computer scientists term it like that, when you don't really need to think a lot for making a code parallel if you have embarrassing parallelism in the code. So this can be achieved over MPI or the internet or the cloud or anything. A typical example for this embarrassing or coarse-grained parallelism is the MPI version of RaxML or RaxML-NG, where, for instance, you just do distinct maximum likelihood tree searches that start on different starting trees. So you have like a master process that is orchestrating those computations. Say you want to compute 100 maximum likelihood trees, and then it just has a couple, like a pool of machines available, and tells each machine to start inferring one maximum likelihood tree. The other application, so this is just for tree searches, for doing independent tree searches, but you can also use that for calculating confidence values. So of course you know that trees without node confidence values are mostly useless, And under maximum likelihood, we do non-parametric bootstrapping. And in the Bayesian world, we do a metropolis-coupled Markov chain Monte Carlo, where we also have independent Markov chains running and independent runs, maybe. So the heated and the cold chains, and every once in a while they're communicating with each other. So if we look at bootstrapping, I'd assume you know that, but nonetheless, so we have this original alignment. Then we just draw sites at random with replacement from this original alignment. We do the perturbation, and then for each of those bootstrapped alignments, compute a tree. And so this needs to be done maybe 100 to 1,000 times, and is an embarrassingly parallel problem, right? Because those bootstrap tree inferences or the bootstrap replicate alignments are totally independent from each other. And then here's another example about search strategies for maximum likelihood analyses. So this is our tree space here, with the bad trees located down here, and the absolute maximum likelihood tree located up here that we're never going to reach just because the search space is so large. And here we may just start two independent searches through this search space indicated by the two arrows here, on different machines. The way this works with MCMC is kind of somewhat similar, only that we have like the heated chain and the cold chain that is shown in green here, and then occasionally they may synchronize to swap chains, then move on to this desired area of high posterior probability. Okay, then the second level of parallelism is what we call inference parallelism. So here we would actually try to really parallelize the search algorithm. This is typically also done with MPI. The problem is that this is not like a general parallelization scheme, but really algorithm dependent, right? So how you can parallelize your algorithm, the search steps by themselves, actually really depends on how the search algorithm is set up. So this is not very interesting because this is really algorithm dependent and very specific. And typically because of the way our search algorithms are designed is also very difficult. And then finally, so the interesting part or the interesting level of parallelism is loop level parallelism. So here we can use all sorts of technologies, OpenMP, pthreads, GPUs, clusters with fast interconnect. We could even use programmable hardware called FPGAs or the Intel Xeon Phi architecture. So this is the interesting level of parallelism, the one we're going to talk about in more detail. So if we look in a very abstract way at our calculations, we always have this configuration here where we want to compute the conditional likelihood vector P given the conditional likelihood vectors Q and R, and essentially every entry I in my conditional likelihood vector P is just some sort of arithmetic function applied to the respective entries I at vector Q and I at vector R. And so the important thing to know is that this operation, like filling the conditional likelihood vectors, uses more than 90% of total execution time, both in maximum likelihood and Bayesian inference tools. But the good thing here is that the calculations of positions I and I plus 1 are independent from each other. So we can apply like a simple fine-grained parallelization to this, right? This is what I said before, that, well, if I have an alignment with 1,000 sites and 1,000 cores, each core can just do the likelihood calculations on one single site. So the way we do that in a very, well, relatively naive way is that we split up the conditional likelihood vectors into equal halves and then have like a blue part of the data and a red part of the data, and then this red thread does the calculations on one half of the data and the blue thread does the calculations on the other part of the data. Right, so one half of the calculations are assigned to the blue thread and the other half are assigned to the red thread. And so just to show you an example how that would look like in OpenMP. So assume, well, we just have this for loop over the alignment, and then at each position of the vector we do this operation where we compute the value of our parent vector p at position I by arithmetically combining the values of the two child vectors q and r at position I. And so here clearly we see by looking at this loop that iterations I and I plus 1 can be computed independently of each other. So if we use OpenMP, we just tell OpenMP that, well, this loop can be parallelized over threads, and so we just add this pragma parallel for loop. And basically that's it. So it will automatically parallelize this loop for us, generate the threads, and split up the iterations over this vector to the threads. Right. And so in practice, what this looks like is that we have some other code. So this may be the sequential portion of the program. Then we have this pragma parallel for. So we've updated our conditional likelihood vector p, and then it is followed by some other code. And the way this looks like is as follows. So we have like this single thread that is doing the sequential operations until we enter this parallel region. Then in this parallel region, 2 or more threads are started, and then once we exit the parallel region, so this is what we call a parallel region, the threads are synchronized again, so we make sure that all threads have finished their work and we have valid entries in our conditional likelihood vector p. And then, so basically here I'm just showing you again how the work among the threads will be split up in the most simple case. And then the sequential thread, the single thread, is going to continue executing the sequential part of the program. Okay, so here's like just another way of visualizing how the data will be distributed among CPUs and also the cache memories. Don't forget these when I'm using this loop level parallelization. Right. So here we still have, in this very simple example, here we still have a parallelization or synchronization point here after the update of every conditional likelihood vector. But actually we don't really need that. We need less synchronization points. So here again is our post-order traversal of the tree. And now, once again, we're splitting up the data among 2 threads. And the question is like, when do they actually need to synchronize, right? So in the simple OpenMP code I've showed you on the previous slides, we need to synchronize 1, 2, 3 times after each conditional likelihood vector update. But this is actually not necessary. So what we can do here is to have each thread traverse a copy of the tree topology completely independently. Then if it reaches the virtual root, compute the sum over the per side log likelihood of the sites that have been assigned to it. So the sum over the per side log likelihoods of the blue part of the data, and the sum over the logarithms of the per side likelihoods of the red part of the data. And then in the end, they only need to communicate once to compute the sum over those 2 partial sums. So this is just like one summation operation to obtain the overall log likelihood score. So we can actually, if we do it in a little bit more involved way, we can actually avoid a lot of synchronization points. Okay, so what about the scalability of those codes? How many cores can I actually really use? So if we have data sets that look like this, alignments that look like this, so few sites, 10,000 sites and 55,000 taxa, we get bad scalability. Because I cannot really distribute those 10,000 sites to that many cores, maybe to 10 or 20, but if I do more, if I distribute it to more cores, the synchronization overhead and communication overhead will be so large that you will not get any further speedups. Whereas in the other case, if we have typical phylogenomic broad data sets, let's say 100 taxa but 1 million sites, we have good scalability. So here I could easily use around 1,000 cores to conduct simultaneous likelihood calculations on such a data set. So the rule of thumb is that, at least for our codes, that every core should work on 1,000 DNA sites or more, so 1,000 alignment sites or more, for attaining good scalability, good parallel scalability and good parallel efficiency. Right. A couple of more notes about likelihood parallelizations. Distributing the data evenly among cores is not that easy for partition data sets. So back in 2016, so I will not go into the details, I will just mention it. So basically back in 2016 we did like a very theoretical paper, algorithmic paper, about how we actually need to distribute the sites of a partitioned alignment to different cores to attain good efficiency. And so far what we've assumed always until this point here is that, well, if you go back here, we're always splitting this in half, right? So this means that we're assuming that every site has the same computational cost. But if I apply some optimizations to the likelihood function, if the cost of calculation per alignment site varies, it's not that easy. So we've, in the meantime, we've produced like 4 papers dealing with this. So the first paper, or like 3 papers dealing with that. The first paper is efficient detection of repeating sites to accelerate phylogenetic likelihood calculations. So this basically looks at different sites and how their similarities to detect repeating patterns in those sites, and that thereby omit unnecessary or redundant calculations. And then this generated a lot of data distribution problems with which load balance, parallel load balance problems that we're currently still fighting with. Just to keep in mind that, in principle, the parallelization approach looks very easy, but if you have partitioned data sets, it gets more complicated, although this is essentially solved because we found a very nice algorithm that solves the problem. And if the cost of computation per site actually varies, so the calculation cost between sites is not independent anymore, then it gets really complicated. Okay, and so here is a small advertisement section, our codes for supercomputers, the ones that we have developed. So one is Exabase, it is similar to Mr. Bayes, but on DNA data sets it's approximately 30 times faster, but it has the same kind of convergence properties and quality of the results, but much better parallel scalability. So the focus of Exabase was really to develop a Bayesian code with extremely good parallel scalability. So for instance, we executed it on a data set with 200 taxa and 100 million sites using 32,000 cores on the Munich supercomputer. So this is a scalability plot of Exabase. So here you can see the speed up over the number of processes, and here you can see the parallel performance, like the parallel efficiency. So it's doing like a weird upwards jump here, which is probably associated with cache efficiency. And here, so this is a very interesting plot. So this is the parallel performance of RAXML compared, or the parallel efficiency of RAXML compared, so this is the blue line here, compared to XML, which was our previous maximum likelihood tool for supercomputers. So here on the x-axis you see the number of cores we used for 2 very large DNA and protein data sets, and this is the parallel efficiency here on the y-axis. And what is very surprising here is that for for 512 cores on DNA data and for 64 cores, but also for other core counts on the protein data, we're getting more than 100% parallel efficiency. And this is theoretically not possible, but what you're seeing here is this effect of increased cache efficiency, right? Because the more cores we use, the larger our accumulated cache memory is that is available. And as maximum likelihood or likelihood-based programs do a lot of memory accesses all the time and don't reuse their data that much, the cache efficiency really plays an important role. So you can see here that for protein data, we even achieve parallel efficiency of 140% just because the accumulated cache memory is so much larger. Right, so basically what is suspicious about these plots, this is what I just discussed. And here, like if you prefer using IQ-tree, here's a comparison of the parallel efficiency of RAXML over compared to IQ-tree. So you can see here that IQ-tree had a different focus, so the parallel efficiency, they never really focused that much on parallelization, so the parallel efficiency of IQ-tree is substantially lower. Right. More comparisons. Well, in the tests we conducted, RAXML found the best scoring tree most often, like for 19 out of 21 data sets. It's between 1.3 and 4.5 times faster. It does have a parallel efficiency of up to 120%. And so, in general, RAXML is faster and returns higher scoring trees on taxon-rich MSAs, meaning on data sets with a huge number of taxa, just because of the design of its search algorithm. On the other hand, IQ-tree results exhibit lower variance. So if you do multiple tree searches with IQ-tree, you will have a smaller variance among the maximum likelihood trees. So conclusion here is that for multiple sequence alignments with strong phylogenetic signal, meaning broad alignments, IQ-tree may require fewer searches than RAXML. Right. So here's the respective publications, and by the way, the IQ-tree guys are our friends, so we've known them for, I think, over 15 years or something like that, and are frequently in touch with them about various implementation issues. Okay, another tool that we've recently developed is a tool called Part2. It's a tool for massively parallel model selection and phylogenetic tree inference on thousands of genes. So this is something that you want to use when you're doing like gene tree, species tree reconciliation. You have a large data set with 1,000 or 2,000 genes, and for each gene you need to do like a maximum likelihood based tree inference. So the cool thing here is that Part2 will do that automatically for you. On the one hand, so it will run a model test and do maximum likelihood tree inference, will do bootstrap inferences if you want to, but as the genes vary in size, right, so not all genes have the same number of taxa, but they also do not have the same length, we have this very nice load or data distribution algorithm where you can just launch Part2 on 512 cores, for instance, and then for each gene individually it decides with how many threads this gene should be analyzed using model test and RAXML. So here you can see that only in this, so this is basically, those are the cores, this is the execution time, and those blocks represent the execution time or the analysis time for a single gene and the number of cores that have been used for analyzing this gene. You can see here that only in the very end we enter this gray area where not all cores are fully utilized, but we still get like something like a core utilization efficiency of around 95% with Part2. So this is very useful if you want to do like a species tree inference based on gene trees with Astral or something like that. And then finally we have GeneRax, which is a tool for correcting gene family trees given a species tree, something is missing here, under gene duplication, gene loss, and lateral gene transfer. So here again we have like a parallel version of that. So this just was published in Molecular Biology and Evolution maybe a month ago. We once again have parallelized this. So this is the linear speed up, and this is the speed up of gene racks, which we executed on a cyanobacteria dataset with 1,099 gene families, using between 4 and 512 cores. So there's another scalable tool if you want to correct your gene trees, and if you want to infer like gene duplication, loss, and lateral horizontal gene transfer events. Okay, so this is our GeneRax tool compared to other tools that have been parallelized with respect to the parallel efficiency. Okay, so now let's soon start with our final section on obtaining CPU time. Okay, so let's talk a little bit about obtaining CPU time on CPU time grant application. So typical system you would apply for is a Munich supercomputing system, the SuperMUC, on which we already have accounts. And this is a little bit, I'm talking about this a little bit from the perspective of the steering committee member, being a steering committee member of the Munich supercomputer, where basically my main job is to coordinate the review of CPU time applications. So what do you actually need to get access to such a large supercomputer? What you need is what we call a closely coupled parallel code that uses the interconnect and the cores a lot. So key thing about those large systems is that they don't only have like a huge number of cores, but they also have a very expensive interconnect such that all those cores can work together and communicate frequently on large numerical codes. So for instance, what you need, like examples for such codes, are RaxML and Exabase, for which we've shown good scalability on this system. So basically just doing like blast searches or even running, for instance, gene racks that that essentially executes a large number of RaxML instances but is not really tightly coupled may be problematic. So what you need to do is that you, in your CPU time grant application, is that you need to show that your code scales and can use, say, 5,000 or 10,000 cores efficiently. Another thing that you need to do is to calculate runtime, overall runtime, like how many million CPU hours you need, the memory consumption, and the disk space required. And all those things, like, a couple of words about the code you're going to use, a scalability plot so you can get test accounts to show the scalability, and then runtime, RAM consumption, and disk space need to be part of this application. Right, and as I already said, so for getting those numbers, you can apply for test accounts. And of course, you should also have an interesting scientific question, but the key thing really in the evaluation is the scalability. So you need a code that is suited for this large supercomputer and that justifies using or having this very expensive interconnect. In terms of resources, at the national level, at least in Germany, that I know best, we have like 3 big supercomputing centers in Munich, Stuttgart, and Jülich. And then we have this distinction between small grants that are typically less than 30 million CPU hours, which are evaluated directly by the steering committees of the respective centers. So those are the grants that I get to review. And then we have the large grants exceeding 30 million CPU hours that undergo centralized evaluation. At the European level, you have this PRACE initiative, so the Partnership for Advanced Computing in Europe, where you can also get preparatory access and then project access. Here's the web link if you're interested in that. So I'll just give you an example for the calculations that we did when we submitted our most recent CPU time application. So we had 1,600 insect transcriptomes with 4 to 5 million DNA sites and 4 to 5 million protein sites. The problem here was that, well, the data sets were not available yet when we were applying for the grant. So how could we measure runtimes for this? A good thing, of course, is that you know how to calculate the RAM requirements for a data set with this dimension, but still predicting the runtimes is a different issue. So what we did is that we just kind of simulated data sets of this size, right? So we just used simulated data sets to be able to predict runtimes. And here's an example. So we predicted that we would do like 50 maximum likelihood searches on the DNA data, for which the runtime on the simulated data set was 60,000 hours. So in total, this makes 3 million CPU hours. For the protein alignment, just because protein data has 20 states and not 4, the time for conducting a single maximum likelihood search was 400,000 CPU hours. Multiplied by 50, so this was just an estimate, this makes 20 million CPU hours. So in total we have 23 million CPU hours for conducting maximum likelihood tree searches on the original MSA. And then for calculating the RAM requirements, we just used our formula. And typically what you need to indicate here is how many how many RAM in total you need, how much per node, and how much per core. Right. Then typically what you would include is some sort of scalability plot. So this is a exabase scaling on the SuperMUC computer. So this is again the number of processes. This is scaling factor, and this is the scaling performance, so parallel efficiency, which you can see here is also superlinear, so above the theoretical optimum. So some plot like this is actually fine. Right, and so that's the end of this part of this lecture on high performance computing. Thank you very much for your attention.